So let's start with the functions of blood. I will list 6. The first one is transport. Blood is a transport medium. Transported in the blood will be things such as nutrients, metabolic waste products, respiratory gases, and hormones. If a substance is hydrophilic, it will be transported dissolved in the water of the plasma. If it is hydrophobic, it must be transported bound to a plasma protein. The second function is defense against pathogens. This is going to involve your leukocytes or white blood cells, as well as antibodies in the blood. The third function will be to maintain pH. An important buffer system in your extracellular fluid is the bicarbonate buffer system. We will study this in greater detail. But this helps maintain blood pH. The fourth function will be the prevention of blood loss. This is through the process of coagulation and involves clotting factors as well as platelets located within your blood. The fifth function is to maintain body fluids. Solids in the blood help determine the direction of osmosis and therefore where water goes, whether it stays within the blood vessel or goes into the interstitial fluid. The sixth function will be heat regulation. Recall that water has a high heat capacity, which means that it can carry energy in the form of heat. Through the processes of vasodilation and vasoconstriction of the superficial blood vessels, you can either eliminate the heat from the body or retain it in the body. We'll talk about this in greater detail as well, but you did go over this in AP1. Now let's look at the volumes. The volumes of blood, or how much an individual has, is dependent upon the weight and the height of that individual. On average, females have between 4 to 5 liters of blood, and males have between 5 to 6. So I'm going to be using the volume of 5 liters for this course. Now let's look at physical characteristics of blood, and we'll begin with color. The color of blood is dependent upon how much oxygen is in the blood. Oxygen-rich blood is going to be a scarlet or bright red. Oxygen-poor blood will be a dull red. In this photograph, you can see the stark difference between the two. The pH of the blood is going to range between 7.35 and 7.45. Arteries contain more oxygen, veins contain more carbon dioxide. The levels of carbon dioxide influence the pH. The higher the levels of carbon dioxide, the lower the pH. Since veins carry more carbon dioxide than arteries, the pH in the veins will be around 7.35, and in the arteries, it will be 7.45. Blood's temperature is 1 degree higher than your core body temperature. Core body temperature is 37 degrees Celsius, so that makes the blood temperature at 38 degrees Celsius. And that's due to the fact that the water is carrying energy in the form of heat. The fourth characteristic is viscosity, and this refers to the thickness of the blood. Blood will be thicker than water, and this is primarily due to the formed elements or the cells. Do you remember what type of main tissue blood is? Recall that the 4 main tissue types are epithelial, connective, nervous, and muscle. So blood is a connective tissue, and all connective tissues are going to have 2 main components: the living cells and the non-living extracellular matrix. In the extracellular matrix, the protein will be fibrin, which is involved in coagulation or blood clotting. Let's look at the extracellular matrix. It's called plasma in blood and makes up about 55% of the volume of the blood. The living cells are called the formed elements, and they're going to consist of leukocytes, which are white blood cells, erythrocytes, which are your red blood cells, and platelets. The formed elements make up about 45% of your blood, and the majority of your cells will be red blood cells. Over 99% of the formed elements will be red blood cells. This is because leukocytes do not remain within the blood vessel. They are traveling through your tissues. I'm going to briefly give you the definition for the term hematocrit. You'll cover this in lab. This is an important clinical test. The hematocrit is the percentage of red blood cells to the total blood volume
of your sample.
On average, it will be about 45%, since the formed elements make up 45% of your blood. But we are going to see in lab that there are some variations between males and females. If I take a sample of blood, put it in a test tube, and then spin it down using a centrifuge, I can separate the blood into different components. Again, you'll look at this in lab. But notice up at the top, the straw-colored fluid, that's going to be the plasma. And at the bottom, you'll see the red column, which is your red blood cells. In between, there's a very thin line, and this is called the buffy coat, which is going to consist of the platelets and the leukocytes. And that buffy coat will make up less than 1%. We're going to begin with the plasma. Plasma is primarily water. The amount of water is dependent upon the level of hydration, but on average, about 90 to 93% of your plasma is water. In that water, you have dissolved substances, such as nutrients. These include amino acids, glucose, fatty acids, glycerol, cholesterol, and vitamins. A second substance are your salts. These are electrolytes. Remember, electrolytes are charged ions or atoms, and they're going to include sodium, chloride, potassium, bicarbonate, to name a few. The third group of substances are your respiratory gases, and these include oxygen and carbon dioxide. In addition, hormones, waste products. These are metabolic waste products. They include urea from protein metabolism, uric acid from nucleic acid metabolism, and creatinine from muscle metabolism. The last substance that I have here are your plasma proteins. Let's look at those in greater detail. The majority of your plasma proteins are albumins. They make up 60% of the plasma proteins. Our albumins are important because they help regulate the osmotic pressure. In other words, the direction in which osmosis occurs. They are an important solute in your plasma. They also function as transport proteins. Recall that hydrophobic substances have to be transported bound to a plasma protein. Albumins carry out that function. Globulins. They are the second most abundant type of plasma protein, and there are three types of globulins. Alpha and beta are going to be transport proteins, and gamma globulins are going to be antibodies or immunoglobulins. Gamma globulins, or these antibodies, are released by a type of lymphocyte called a plasma cell. The third group of proteins is fibrinogen. Fibrinogen is a clotting factor. Plasma without the clotting factors is called serum. In addition, regulatory proteins such as hormones and enzymes round off the types of plasma proteins that you have.